Go on, GCSE science people. It's GCSE boss here back with another quick one about the photosynthesis required practical. Okay, so in this practical, the aim is we want to investigate to see how the light intensity affects the rate of photosynthesis. Okay, so what happens in this practical? The pondweed is shifted closer or further away from the light source. Okay, and what happens is in photosynthesis, one of their products is oxygen gas. Make sure you know this equation. Now, because a gas is given off, you're going to see bubbles when photosynthesis start occurring. So what we're doing is we're counting the number of bubbles at each different length that the pondweed is placed towards the light source, which therefore means that our independent variable, the one that you are changing, is the light intensity. Okay, so the closer the pondweed is to the light source, the more intense the light, and the further it goes away from the light source, the less intense the light. So that's what we're changing. We're changing the distance, which therefore means we're changing the light intensity. Your dependent variable, which is the one that you're measuring, we're counting the number of bubbles formed per minute or per 2 minutes or for however time that you've placed it at that distance. And your control variables are what you're keeping constant. So we're trying our best to keep the temperature constant by using an LED light source. The amount of pondweed doesn't change, and we don't change our carbon dioxide concentration. As a matter of fact, we use lots of sodium hydrogen carbonate to ensure that there is enough carbon dioxide and it is not a limiting factor. Okay, all right, so the improvements that can be made, you can see that to the side there. Instead of counting the bubbles, because you can form 2 bubbles or 3 bubbles, okay, instead of sitting and counting that, you could use a gas syringe. Also, another improvement is that you could use your phone to record and replay in slow motion and count the number of bubbles. Okay, or some bubbles may be bigger than others, so it's best to use a gas syringe to measure the volume of the oxygen produced. Okay, now this is what your table of results could look like. And if you notice at different lengths here, you're using you're carrying out the experiment 3 different times at a particular length. Now, why do you repeat the experiment at different lengths or different distances? We do that so that we can identify any anomalous result. You never ever say to improve accuracy for a repeat. All right, make sure you know that. Now, all the best in your GCSEs, and please comment and let me know which required practical do you want me to do a quick summary on. This is GCSE Science Boss. All the best in your GCSEs. I'm out.